Thanks again for the invite to speak today, and hello everyone. Today I'm going to be talking about our research in representation learning for computational pathology. I'm a professor at the University of Utah in the Electrical and Computer Engineering Department and the Scientific Computing and Imaging Institute. Okay, so for those of you who might not be so familiar with this, just a very quick kind of background on this. So the typical clinical workflow for histopathology is obtaining these hematoxylin eosin stained whole slide images. These can come from biopsies or they can come from tissue resection in a surgery. And typically these are very large images on the order of hundreds of thousands of by hundreds of thousands pixels, and they are composed by staining the tissue with two different chemicals, hematoxylin, which stains the cell nuclei and has this bluish purplish color, eosin, which stains the cytoplasm and has kind of a pinkish color. And the image is, of course, captured as a color image, which is a combination of these two stains. Just for reference, some smaller, more computer vision workflow size tiles are shown on the right, examples of normals and examples of cancer patches, and these are usually on the order of 256 by 256 or thereabouts pixels. So the workflow goes something like we get the whole slide image and we then crop it into these smaller patches, right? And these can be in the thousands or up to even 10,000s. And these are first processed individually. Nowadays, there are many great foundation models that are trained to encode these patches into high-dimensional features. These are typically now transformer-based models. And then, doing a classification at the level of patches is pretty difficult in this application space because, as you can imagine, it would take a lot of effort for a pathologist to go ahead and try to label all of the patches that you extract from a whole slide image, and especially considering large data sets are going to have thousands maybe of whole slide images, right? So the typical workflow in histopathology usually uses slide level training labels, which can be extracted readily from reports, right? And then keeping the patch level encoder frozen, typically an annotation-based slide level classifier is trained using multiple instance learning. So having said that, I want to try to see if we have time to focus on three different aspects of our research today. So I want to ask several questions. First, can we improve patch level classification? Is that possible? Can we leverage semi-supervised learning? Then I want to move on to can we improve these patch features for specific downstream tasks over what foundation models, the more generalist models, can provide us? And finally, I want to discuss if we can improve task accuracy for tasks that might need information beyond just patch level features, like more about tissue architecture. So to get into this first part, I'm going to introduce this contrastive loss we've used for in conjunction with stain separation for semi-supervised learning at the patch level, right? Okay, so I already mentioned that it's near impossible to exhaustively kind of label patches from these huge images, but sometimes what we can get from a pathologist is them looking at this slide at kind of a moderate resolution, perhaps zooming into parts and zooming out, and then kind of outlining areas of cancer, areas of normal tissue architecture, right? But by no means is this like an exhaustive labeling of the slide. So if something is not annotated, that doesn't mean that it's not cancer, that it's normal. It just simply means that they didn't look there or they didn't get there, right? So we have a lot of unlabeled data, a lot of unlabeled patches, and maybe some examples of, for instance, if the problem is cancer versus normal, some examples of cancer patches and some examples of normal patches. So of course, there are a lot of semi-supervised learning methods in the computer vision literature, but the question we wanted to ask is, can we build something that's specific to pathology that might kind of leverage the particular properties of these images and kind of outperform the more general methods? So what we're building on is this, very well-known decades old approach called co-training, which relies on multiple views of samples and applies some sort of consistency kind of loss between the predictions from multiple views. So it fits well to this problem because, as I already mentioned, these images are composed of two stains, hematoxylin and eosin, and they represent slightly different things, the nuclei versus the cytoplasm, right? And also there's been a lot of work, existing work on taking the RGB image and decomposing it back into the H and the E components, right? So that's what we start with. We take the RGB image and decompose it into its H and E channels. Then we encode each of these. Back then we used a ResNet. So we have a ResNet, same architecture, but different weights, one to encode the H channel, one to encode the E channel. Then we apply a contrastive loss in this feature space, right? So the particular loss we used was a triplet loss. So we have positive pairs and negative pairs. The positive pairs we draw from the H and E channel features of the same tile, same patch. And we can get negative pairs from H and E channel features of non-matching tiles, so different tiles, right? And the idea, without going into too much detail, is to pull together the features of the positive pairs in the embedding space and push away the features of the negative pairs. And of course, since contrastive loss here does not need a label, it can be applied to the whole set of patches extracted from whole slide images, whether they're labeled or unlabeled. Then in a typical semi-supervised learning fashion, we combine this with cross-entropy loss on a much smaller labeled set of patches, right? And so my student was able to show that this actually does outperform in initial experiments, that it outperforms kind of these more general methods, which were kind of state of art back then, fixed match, mixed match from computer vision. Now, we also wanted to ask, understand a bit more why this works, right? So, for instance, this idea of co-training and the contrastive loss between the two channels, could I simply take the red and blue channels from the image, for instance, forget about the H&E, apply the same idea there and get the same gain? Turns out, so we tried this and we expected that it would not work, and it indeed did not work. We did not see improvements in this case when we used unlabeled data for semi-supervised learning. Turns out in co-training, one of the main assumptions that you make is that your multiple views are somewhat conditionally independent of each other, right? And we thought that H&E channels fulfill this, whereas the RGB channels do not. So to kind of also get more evidence on this, we did this experiment where we took the H channel, for instance, and used the UNet, trained the UNet to try to predict the E channel and vice versa. And we saw some moderate kind of prediction accuracies here. The R-square value in these tasks were about 0.5, right? So very moderate kind of fit. On the other hand, if you take pairs from the red, green, blue channels, the RGB channels, then and do the same exercise, you see much stronger prediction accuracy. So there's a lot more correlation in the RGB space than the H&E space. So that's an example where we are kind of trying to leverage the particular kind of properties of the H&E images to our advantage. Then my student went on and combined this with this loss with multiple other kind of losses, more general things, pseudo labeling with sharpening and mix up, and kind of tried to get the best results he could, and kind of demonstrate. I don't want to focus too much on these results in the interest of time, but he used one data set from our institution and then the TCGA kidney cancer cohort to show that this indeed does better than some other alternatives that are out there and the ablation studies. But in the end of the day, the advantage of this patch level approach is you can kind of get this nice kind of semantic segmentation like outputs, right? So not just predictions for the whole slide, but actual localized predictions. So moving on to the second part then. So going into now the more kind of common workflow where you don't do patch level predictions, but do slide level predictions. The question, again, to remind us is, can we improve patch features for downstream tasks over what generalist foundation models might give us? Right? So for this part of the talk, I want to, again, briefly start with a little bit of a background, what this problem looks like, right? So we have two whole slide images here. The one on the left is an example where there's cancer. The one on the right is normal tissue. And the particular way the slide level labeling is going to work here is that, so if we call the slides, the whole slides, a bag, a negative slide, that's a normal slide. by definition will not have any cancer in it, right? So if I look at any patch from this slide, I know that they're all going to be negative. But on the flip side, if I look at the left picture here, the cancer, so okay, if we know that the slide, is cancerous, that does not mean that every part of this tissue has cancer. There's going to be a lot of normal tissue there as well, and there's going to be some cancer patches in there. We are not going to know which one is which because the histopathologists, they don't have time to go ahead and do this labeling as mentioned already, right? So all we know is that there is going to be cancer patches in there. Okay, so, and the typical workflow again looks something like this. So the first step, we crop the whole slide image into patches. We encode all of those patches using our favorite foundation model, for instance, and then that is frozen. And in step two, a separate multiple instance learning strategy is used to then take these features from the patches and output slide level classification results. And as mentioned, this is typically kind of a simpler attention-based aggregation followed by some classification, maybe like an MLP, though certainly there are more complex models out there that use transformers to do this as well. But it is limited by what data is available. Whole slide images are large, so we have lots of patches in these problems, but the number of whole slide images themselves are usually going to be limited to maybe a few thousands or maybe tens of thousands if you're really lucky, right? So that also limits how complex you can make that step two. I mentioned this because when you think about that in combination with the fact that the foundation model is frozen and the patch level features are frozen, if those features are pretty entangled between the things that you're trying to separate, for instance, normal versus cancer, you're going to have a little bit of a hard time at that step two because the model there is not so powerful and the features are entangled, then it's going to have a harder time, right? On the other hand, if the features are disentangled for your particular task, then that second step is going to have an easier time. So that in this part, essentially, we're asking, can we do this? Can we utilize slide level labels to pre-train the encoder so that we make the task easier for that second step? All right, so just a reminder, I'm sure many of you already know this, but I'm going to talk about two kinds of contrastive learning. The first is the very commonly used self-supervised approach. And obviously there's many different precise formulations of this, but the general idea typically is I take an image that's called an anchor. I create some positive views of it, some other augmentations of the same image. So I know they belong together. Then I sample some, maybe randomly some other images, which I call negatives. And what I want to do is, use a loss function that essentially forces the model to map that positive pair, map the positive pairs close together in the embedding space and the negative pairs far away in the embedding space, right? So this is the bottom line usually. So on the right here, I have this kind of exaggerated illustration, right? The kind of space you might end up with here, where you have different views of the same objects clustered very close together. But when you look across the different classes, right, cat versus dog here in this example, they might actually be kind of entangled to some extent, right? Kind of going back to that previous point. Now, a second kind of contrastive learning is supervised contrastive learning, less commonly used because clearly it requires labels and it does not serve well as a pre-training task in many scenarios. But here, what we could do is also leverage the labels of the images to enlarge, for instance, the set of positive pairs. So not only will I take the same dog and create views of it and call them positive pairs, but I can also sample images of other dogs and call them positives with this first dog, right? And when I do that, and I use a loss function then to pull those positives together and push away the negatives, then we end up with a different kind of representation space, right? Different kind of embeddings, where now we can expect the different examples of the same class are going to be clustered together, and the clusters between different classes are going to be better separated. Now, depending on what you want to do, this might or might not be a good idea, right? So if you're after training a foundation model that's going to be good for a large number of downstream tasks, clearly this is probably not a good idea because you're going to lose a lot of important information, right? You're not going to be able to distinguish between a yellow cat and a gray cat here because you've lost that. But if your goal downstream is to separate cats and dogs, then maybe this is not such a bad idea. Okay, so now we're going to map these two things back onto our problem, right? So remember, we have the negative negative bags, where we know all the patches are negative examples. And then we have the positive bags where we don't know. We know there are some positives in there, but there's most likely a lot of negative patches in there as well, right? So the idea, the algorithm is the following, right? So when we form batches and for our learning, we crop patches and then we pass them through our encoder and our projection head. And then when it comes time to apply the loss function, then we separate out the two kinds of patches we have. We have the patches from the negative bags, which we know they must all be negative. So here we're going to apply supervised contrastive learning. Okay, so the similarity loss here means an implementation of supervised contrastive learning for the negative cases. And if the patches are coming, if a patch is coming from a positive bag, then we don't know if it's a negative or a positive. Here, we're going to use the self-supervised contrastive learning, right? So what we can kind of expect is a picture like this if we think of the two parts together. So on the bottom left here, like I can expect that my embeddings from the negative patches are going to cluster together pretty tightly. On the other hand, the embeddings from the patches from the positive bags, they are not going to cluster together very tightly, but I can expect that different views of the same patch are going to be clustered together. So when you put these two together, though, there's one more thing that comes into play, right? The negative patches, so the normal parts of the tissue in the cancer site, so the negative patches in the positive bags, actually look like the negative patches from the negative bags. So they're going to be encoded similarly by our model, right? So in the middle here of our representation space, we expect kind of this overlap between negative patches from both negative and positive bags. And then kind of further away, we expect the positive cases. Okay, so we actually, after training our model, like in different ways, we visualize the features to kind of confirm that this is indeed happening. So, and we see that more or less it is, right? So this is for the Chameleon 16 data set, which is lymph nodes from breast cancer. And on the left is the self-supervised, just purely self-supervised learning in this data set without paying attention to the label of the bag, and then doing a 2D PCA visualization. And we see a lot of overlap here, right? Now, this does not mean that these features would be useless for downstream tasks, but because they're in high dimensions and there's probably some separation there, which a powerful classifier could use, but it's not a very simple task either to separate these. Now, on the other hand, if you apply the supervised contrastive learning indiscriminately to both the positive and the negative bags, that's the middle column here. At training time, you might think that, oh, I'm doing a good job, right? Because you're overfitting essentially to your positive and negative bags, pretending like everything from the positive bag are positive, but that's not true. So at testing time, you see that again, there's a lot of overlap between the features. So on the right-hand side, this is the approach that we took, right? The weak SUPCON approach, where you apply the supervised contrastive loss just to the negative bags and the self-supervised contrastive loss to the positive bags. Now, both at training time and to a large extent at testing time, you see a better separation between the positive bags, positive patches, and the negative stuff, right? So you still expect some overlap between the red and green here because remember, there is actually negative stuff in those red dots. Okay, so then my student went on and tested this on 3 different data sets. One is Chameleon 16, as already mentioned. This is a public monthly institutional data set. And then 2 of our kind of institutional data sets here at the University of Utah, renal vein thrombosis and kidney metastasis. So notice that the numbers here, like our data sets are much smaller in the number of cases. So each case might have multiple slides, but so we have, a few hundred maybe whole slide images here to work with, right? So, okay. So looking at the results here and comparing our approach weak SUPCON to the kind of the more established approach of taking a foundation model in this space, so for instance, Unit 2H, right? And treating this as a frozen encoder, training your mill, and doing your pipeline this way. On Chameleon 16, the publicly available method, that approach, the more traditional approach, typically outperforms us by a little bit, right? And that's kind of expected because those foundation models are trained on a very large number of slides, and for public data sets that are out there, they do a great job. They do okay job for most private data sets also. For instance, on the kidney metastasis data set at the bottom here, they were like our method is pretty close. Maybe it's a little bit better than this approach because the features themselves are better tuned to the problem. But then in the middle, so we have the RVT data set where we were surprised, in fact, to see such a strong difference that we by quite a large margin, we were able to outperform the foundation model features. So, this could be because maybe this data set is somehow, out of distribution for the foundation model. It hasn't seen examples that are similar to this, right? So this gives a case where you can actually, it might be beneficial to use a pathology specific kind of idea like this and, apply it to your own data set to train a model either from scratch or to take a foundation model and then fine tune it, right? So, okay. So finally, moving on to the third part here. Last question, can we improve accuracy for tasks that need information beyond patches, right? So most of these mill methods treat tissue patches as unordered bags. They discard the spatial layout. And this is acceptable for a lot of tasks such as cancer detection or grading, where like mere presence of a specific cell population dictates the outcome, right? So if you see these bad cancer cells, and you know that this slide has bad cancer, right? But it's suboptimal for some other tasks, maybe a little harder tasks like distant metastatic risk prediction, okay, which relies on geometric arrangement of the tumor microenvironment, right? So what do I mean by this? For instance, if somebody gets tissue resected in surgery, okay, they are diagnosed with prostate cancer, can we predict if it's already metastasized just from looking at the primary tissue resection? Has it metastasized somewhere else without collecting additional data? If we could do this, this could actually improve timeline to treatment. But this is not a simple matter of finding cancer on that slide because there is going to be cancer, it's prostate cancer. But the question of metastasis maybe needs clues like, is that cancer close to vasculature in this slide? So the geometry of tissue architecture becomes important. And that remains a pretty hard challenge to this day. And unfortunately, absolute coordinate-based positional embeddings are not very meaningful in whole slide images either because the slide architecture, the high-level architecture is very variable compared to the objects we see in computer vision. Okay, so our idea here essentially is to inject this geometry information to the whole workflow, right? So what we do is we take the patch features that are embedded from a foundation model perhaps, and then we do some clustering in that space. This could be k-means type of very simple clustering, or it could be doing zero-shot semantic mapping using a VLM in the pathology space to different tissue types, right? So that's what this bottom middle picture is showing, the different colors here. The 4 colors are the 4 kind of clusters, the 4 prototypes, right? So once we have kind of segmented the whole slide at the patch level into these different clusters unsupervised way, then we compute sign distance functions everywhere in that whole slide to the different clusters, and we derive some additional features from those clusters. And then what we do is at the patch level, when we're looking at a patch, we not only use its features from the foundation model, but we augment it with these sign distance functions computed at that location. So kind of to give a bit more concrete example here. So on the left, we're trying to kind of show that actually k-means is not so bad here. It does a pretty reasonable, semantically meaningful job, right? So if we take the conch model and we use prompts to kind of segment patches into different pathologically meaningful classes here, we kind of see that they actually overlap quite a bit from what we get from k-means. Okay, so Then, taking one of these clusters, for instance, I believe in this case it's the orange one, we can compute a sign distance function to that cluster. And then we apply radial basis functions of different spreads, and we look at the gradient of the sign distance function and some statistics, right? So if we are talking about a patch here, we're going to take its foundation model features, then augment it with these features that geometry aware, slide level features that we've computed. And then we're going to pass it on to the mill, hoping that now we can leverage the tissue architecture. Okay, so we experimented in this with this data set, which is prostate cancer metastasis. And this is a data set from our institution, from the VA, actually. And what we do see here is that indeed, this idea of injecting geometry-aware features does improve this task quite a bit, right? Because as we hypothesized, I think it is important to look beyond just patch-level features and aggregate patch-level features here, and to actually look at the tissue geometry, right? And then other tasks, I'm running out of time here, so I'm not going to go into any details here, but depending on difficulty of the task, that might or might not benefit, right? So for instance, for tasks just like cancer detection, Chameleon 16, where the cancer is already in that slide, then we don't see a benefit from adding these features, right? And that's kind of as expected. Okay, yes, so I just want to end with acknowledgements. The students who did this work, Bodong, Hamid, and Sandesh, and my collaborator in pathology, Dr. Knudsen, and other collaborators and funding agencies who supported us with this work. So thank you for your attention.